Muchísimas gracias por la invitación a este evento. Para mí es un placer poder estar con todos ustedes.
Thank you for your invitation to this symposium. It's a great pleasure to speak to you all about the microbiome of breast milk and the influence in the development. We all know that maternal microbiota is one of the most important factors to determine, to impact the child health. So any early exposure during gestation and the breastfeeding life stage will have a direct impact on the development of the child, and in parallel will have an effect on the programming and development of the immune system and the metabolic activity. We cannot define what a healthy microbiota is, but we can know that children that were born through a vaginal delivery term and following the exclusive breastfeeding patterns as recommended by the World Health Organization are associated to a lower risk of developing diseases. Therefore, we consider this is the standard microbiota. This is a very complex process which starts at the beginning of our lives and evolves in composition, in quantity and diversity of species from the very first life stages over the infancy, childhood, adolescence, so as to become more or less stable when we become adults at 15 to 18 years of age. So different factors such as the type of delivery, exposure to antibiotics, how and when complementary feeding was introduced, exclusive breastfeeding habits, etc. All those factors will play a decisive role and will impact on the good health of the infant. So over the first early life, in parallel to this process, we have the development and maturation of the immune system, cognitive development, motor development of the child. So any effect that can modulate or modify this colonization process, the colonization of the gut, will have an impact both on the immune system and the neurocognitive development, and therefore the health of the child. We know for sure that there are certain bacteria that include short-chain fatty acids, vitamins, and some compounds such as neurotransmitters, polyamines, and other metabolites that play a biological role on newborn babies. We know that we inherit bacteria from our mothers, so they are pioneers, they are the initial colonizers of the gut. Those bacteria play a very important role in regulating and modulating gut homeostasis. They also play an important role in the maturation of the immune system, modulation of the carbohydrate, lipids, and proteins. They help in cell proliferation and differentiation. They play an important role in blood coagulation, etc. So they all have a direct impact on the development of the individual. There are more and more evidence that suggests that any impairment, any alteration in the microbiota are associated to a higher risk of disease. So several problems such as allergies, inflammatory problems, obesity, overweight, celiac disease, autism, and diabetes, etc., are related to impairments, alterations in the composition and the diversity of human microbiota. So let's see what's the impact of the development of maternal microbiota on the development of the child. Some bacteria are transferred through in the moment of delivery and also during the lactation period. So we need an adequate milieu so as to create the proper imprint or trace in the newborn that will have a direct impact on his or her health. We enter into contact with bacteria in 3 times. First of all, during gestation, there is a lot of debate about this, so this is not today's topic. Secondly, the second moment of contact, the delivery. When there is vaginal delivery, there is a great exposure to bacteria because the baby gets in contact with bacteria present in the vagina. And thirdly, we will focus on the third moment, lactation, the breastfeeding stage. This bond, this created between mother and child, will have a direct impact on the development of the gut microbiota. Several studies indicated that both the mode of delivery and breastfeeding habits play an important role. This study, published in Science Translational Medicine some years ago, indicate that mode of delivery can be modulated by the breastfeeding habits. So if the child is breastfed, the difference tend to be less notable, less remarkable. So it's suggested that mother breastfeeding modulates the impact of C-sections and especially exposure to antibiotics. There is a lot of scientific evidence about the good effects of breastfeeding. They say that children that were fed with human milk are less susceptible to gastrointestinal and respiratory infections. Most of the evidences are focused on the gut, but there are other niches on the human body that are inhabited by bacteria. Our team of researchers worked on this area, and we managed to describe that the mode of delivery and the breastfeeding habits play a very important role in the colonization of the oral microbiota. So we observed that the mode of delivery, especially exposure to antibiotics, played a very important role on the oral microbiotic, and this mark was observed up to 6 months of life. And from that onwards, we couldn't distinguish any differences. And this could be partly explained by the introduction of complementary feeding, or maybe because the infant is attending nurseries, in contact with more children, etc., etc. Regarding breastfeeding habits, we saw that those children, those babies that were partially breastfed over the first 6 months of life presented a oral microbiota trajectory that was completely different to those children that didn't follow exclusive breastfeeding habits over the first months of life. We also studied the role played by breastfeeding in the microbiota of preterm babies. We saw the effect of breast milk, human milk, donated milk, and formula milk. We observed that the preterm microbiotas receiving donated milk, this profile is very similar to the profile of the child that receives milk from his mother, whereas there are more important differences with the formula milk. This microbiota was quite different, whereas donated and mother milk were similar but somehow different. And this could well be due to several reasons. First of all, donated milk is pasteurized. This treatment reduces or modifies the bioactive compound and macronutrient profile of the milk. And secondly, donated milk comes from mothers that had their children, they had term babies, not preterm. So the composition of the milk tends to be slightly different. Composition of micronutrients and bioactive compounds is different. So breastfeeding will play a very important role in the colonization of the gut. So which factors make breast milk so important, so interesting beyond nutritional aspects? Because this affects health of the infant short and long term. It's not easy at all to work with samples of breast milk. This is a very complex with sample with bioactive complexes, different compounds that is influenced by the timing of feeding, and it changes in the composition from the colostrum. This is the first type of milk we receive when we are born. Then it transitions into mature milk. We also know that breast milk changes within the same day. The composition is not the same if it's taken in the morning or at night. in terms of cortisol, serotonin, melatonin. They change their profile over the days. Some components follow circadian rhythms. Composition of the milk changes according to the feeding times, according to the moment the child is breastfed, or according to what the mother eats. There are some studies that indicate compositions can change from one breast to another. Recent studies suggest that the milk changes depending on whether the child is a boy, a female, or a male. We are learning a lot. We know that breast milk is a model for immune tolerance because the milk transfers antigens and immunomodulating compounds, immunological compounds, hormones, and also HMOs, bacteria and metabolites. All these will regulate the immaturity we have in our immune system at birth, and this will foster the adequate milieu. So in short, breastfeeding, apart from satisfying from meeting nutritional requirements of the infant, it provides a great variety of immunoactive components, and I would like to stress the very important variety of bacteria that are transferred from breast milk. And with all these components, these are the first colonizers we receive from our mother at the moment of delivery, with direct effects, as we said, in the development of the immune system, protection against pathogen infections. It supports gut homeostasis, and it reduces the risk of having intestinal diseases. Breast milk is a constant source of microorganisms. We used to think that it was sterile up to 2003, but thanks to a very important work from Spain, we managed to demonstrate that the breast milk contained very important bacteria, such as lactobacillus and bifidobacterium. Thanks to the development of new technologies, for instance, massive sequencing, we've increased our knowledge, and around 800 different species of bacteria have been described mostly from Staphylococcus and Streptococcus, although we also find lactic acid, phylobacteria, and also protobacteria such as Escherichia coli and Klebsiella, and they are all in breast milk. So the quantity in absolute terms is very, very, very low. This is the colony forming units per milliliter. But if we think that the child receives around a liter of milk per day, this constant supply of my bacteria over a long period of time will play a decisive role. Which are the important functions? Structurally, because it fosters homeostasis, it reinforces the barrier function, metabolic also, because most of these bacteria are capable of producing vitamins, short-chain fatty acids. They help mature the immune system, they prevent pathogens or other toxic components coming from the diet, will be modulate somehow. So as I said, it plays a very important role. The presence of bacteria in breast milk is not exclusive of human beings. We found the presence of bacteria in the milk of other mammals. Interestingly, there is a great degree of inter variability across women and across times. As you can see in the slide, each bar represents a specific time, and each color represents a bacteria group. the profile S6 of one mother has nothing to do with profile S8 of another mother. If we try to compare microbiota of human milk with other milieu in the body, we see that the clustering is quite different from microbiota from the skin, from our mouth, of our vagina. So this suggests that there is a specific milieu, but we cannot forget that we can find other type of microorganisms in breast milk. Our team, the members of our teams, were pioneers in showing the presence of fungi in breast milk samples. This was accidental. We were studying, and we found out a colony, a very creamy sample, and looking it at under the microscope, it was like a yeast, and by using specific techniques, we found out it was a type of yeast. So by using different cultures and different methods, among others, massive sequences, we could confirm the presence of yeasts, fungi, etc. For instance, Saccharomyces cerevisiae, a very common yeast in foodstuffs, was present in the breast milk. So feeding habits and the environment could be one of the main important reasons of the presence of yeast and fungi. Although we can also find viruses, archaeas, and phages. So the complexity and the variety of microorganisms in the human milk is really impressive. So we still don't know what's the biological role they play in the colonization and the modulation of the immune system of the child, and therefore in its health. Apart from the complexity, we know that the same perinatal factors that affect the child microbiota affect maternal microbiota. And this has a reflection on the milk, on the type of milk. For instance, medicine consumption, maternal diet, of course, the lactation of the stage, environment, maternal health, etc., etc. And if we add the high complexity of the breast milk composition, apart from bacteria and other microorganisms, we find other bioactive components such as cytokines, growth factors, HMOs, hormones, proteins, and peptides, fatty acids. As a result, we can just start to glimpse the colossal complexity of breast milk. We need to understand which are the specific factors modulating this composition, which is the interrelation between the different components, and which are the effects on the newborn health in terms of growth, development of microbiota, reduction of infection risks, allergies, asthma development, etc., etc. Therefore, we can say that there is a direct relationship between feeding habits, microbiome, and immune system, and this is related with the risk of illness. So we need to understand the role played by diet and the transfer of bacteria and the role they play in the child biota, because this will help us develop new dietary strategies geared at programming that will support child development and will support nutritional, microbiological, immunological programming over the first 1,000 years of life. And I would like to finish my presentation by thanking my team and all the funding sources that made possible this work in this field of research, microbiota and lactation. I will be happy to answer to any of your questions. Thank you.